epigenetics and cancer have a relationship because cancer is essentially unregulated, caused by unregulated cell division. So if we have lots and lots of cell division occurring inside of our bodies and it's unregulated, it's uncontrolled by our bodies, then essentially we have cancer. So the reasons why, or some of the reasons why this cell division can become unregulated is because a couple of genes which help to regulate cell division may not be expressed or may be silenced when we don't want them to be because of epigenetic factors having an influence on them. So these two main genes that we're concerned with are something called proto-oncogenes. And what proto-oncogenes do is they promote cell division. And our second type of genes that we are worried about here are something called tumor suppressing genes. And these tumor suppressing genes, from the name, inhibit cell division. So we've got two types of genes, one that in proto-oncogenes that promotes cell division, one in tumor suppressing genes that inhibit cell division. So essentially, if we have epigenetic factors that are influencing proto-oncogenes by activating them and having too many of them being expressed, then we get this rapid promotion of cell division, which can lead to unregulated cell division and then cancer. What we could also see is epigenetic factors could undesirably silence too many tumor suppressing genes. So these tumor suppressing genes, which usually would inhibit cell division, so ensure that we don't see too much cell division, by being silenced, they are not able to keep cell division regulated and under control. So both DNA methylation and acetylation, which is part of chromatin remodeling, can have an impact on this. So let me try and plot these out because it's a little bit complex, but if you break it down, it makes it a lot more easier to kind of understand, get your head around, I guess. So if we see reduced rate of DNA methylation, so remember DNA methylation leads to the silencing of genes. So if we see less DNA methylation, we're going to see reduced silencing of genes, which means that proto-oncogenes won't be silenced where they might have usually. They might be activated. So if proto-oncogenes are activated, then that can lead to unregulated cell division. So remember, if proto-oncogenes are being activated, then it can lead to unregulated cell division because proto-oncogenes promote cell division. Alright, so lower rates of DNA methylation in those proto-oncogenes would mean that they are being expressed. We don't want that to happen too much, otherwise they will promote too much cell division. It may end up being unregulated and uncontrolled. If we see increased DNA methylation, so lots of adding methyl groups to cytosine molecules in tumor suppressing genes, we will see tumor suppressing genes silenced. So if methyl groups are added to the cytosine bases in tumor suppressing genes, it's going to mean that gene is silenced, or those genes are silenced, which can also lead to unregulated cell division. So remember, tumor suppressing genes are trying to inhibit cell division. So if these genes aren't made, then they aren't able to inhibit cell division, so we could be at risk of too much cell division occurring, leading to unregulated cell division. If we compare the same kind of principles to acetylation, chromatin remodeling, if we see decreased acetylation, could lead to tumor suppressing genes silenced. So remember that acetylation causes those histone proteins to push away from each other because of their charges, and it spreads out genes, allowing RNA to access the genes. So if we're not seeing that happen, then tumor suppressing genes can't be accessed, they'll be silenced, and once again can lead to unregulated cell division. Because if they're silenced, they're not being made, they can't try and suppress tumor formation. So a tumor is just a massive growth or lump of cells that have divided really rapidly, so it created this lump or this mass. And finally, if we see increased acetylation, will lead to genes being expressed. So it could lead to proto-oncogenes being activated. Remembering that proto-oncogenes lead to or promote cell division. So this in turn could start to see unregulated cell division become more of a risk again. So basically, if these epigenetic factors sort of come about in an undesirable way, or we see certain genes being activated or silenced that we wouldn't usually want to in proto-oncogenes or tumor suppressing genes, then the end result can be greater amounts of cell division to the point where it could be unregulated and uncontrolled. And if we see that occur, then that unregulated cell division can essentially lead to cancer and tumors forming.